Okay, folks, we are on the home stretch. It's our last week. We're almost there. Can you believe it? With Thanksgiving coming so late, it feels like, next week's Christmas, but it's not. Almost. It's finals week, almost as exciting, right? Speaking of finals, when is our final? It's Monday, right? So a week from today. So that is the 9th of December, Monday. What time? 3 p.m. Where? Right here. Now they allocate, 2 hours for a final. You only need the hour 15 like we normally do. It's going to be very, very similar to a regular exam. The cumulative section will be a little bit more heavy because the only new material that we have is this week's material and then Wednesday's online lecture, right? And then the rest of the material is from, cumulative from units 1 through 4. So cumulative nature, maybe a quarter of the exam is cumulative. Okay, so let's talk a little bit about Canvas. So I've spent quite a bit of time emailing back and forth with IT. I've got a guy in IT now that knows me because every semester about this time, I send him my grade book. I've downloaded it, asking why Canvas isn't matching my calculations, and we figure everything out. So Canvas should be pretty accurate with the following assumptions. You ready? All 4 of the exams that you've taken are counted. I haven't dropped any of the exams because I know some of you are trying to decide, do I need to take the final? All of the quizzes, except for the one that's due Friday, unless you've already taken it. But I've already dropped your 2 lowest in Canvas. Does that make sense? So if you take the quiz that's due Friday, right, we've got one final quiz due Friday, quiz 10. So if you get that done, Canvas will populate. It'll automatically drop your 2 lowest quizzes. Then the participation points for Unit 5 are not there. You will need to have those. You'll need to consider those. There's, right, 10 more points in participation. You don't want a 0 out of 10. So if you're thinking like, hey, my grade looks pretty good, just come to the rest of class this week, get your participation points, attend a review session. We're going to talk about review sessions in a second. You'll get the last 5 of participation points. You don't need to do the bonus number 5 because it won't count. It only counts if you take exam 5. And then maybe email me and let's just confirm, or come into office hours and let's just calculate it before you just like peace out, don't show up on Monday. Okay? But if this is my Christmas present to you, right? If you've worked really hard all semester and you're going to count all 4 of your exams to date and you're going to drop your lowest, that could be the final. Merry Christmas. You don't need to take the final. You don't even need to show up. I do want you coming to class. That's why the participation points. And I want you going to review session. You just don't have to perform on the exam. Or you can come and just see how you do. Okay? Maybe just see how you do. But focus on another class that's maybe a little bit more precarious. Does that make sense? I want you coming to class and I want you going to the review session for those points because you need to be exposed to this material. Like, this material is important. Even if you don't take exam 5, you still need to know it. Okay, so I don't want you to completely blow it off and be like, a patient comes in with an eye issue, a retinal detachment, you're like, well, I dropped that exam, so good luck, right? You're on your own. Any questions about that?
Will there be a study guide for like this unit?
It's already up. Study guide's already up. At the end of the semester, after all of the drops have happened and everything is tallied, and you have a final grade of an 89.49%, what is your final semester grade? a B. Do you remember the first day of class when we walked through that? It's right on the syllabus. If you have an 89.5% or 0.6 or 0.7 or an 89.9, what is your final semester grade? An A. If it's an 89.49, I get it. I feel for you. But I have to have drawn the line somewhere. So I draw it at the beginning of the class. I stick with it at the end of the semester. I feel that's the fairest way to exercise it. If you reach out to me, I will repeat that in the nicest way possible, but that essentially will be the message. Okay? Dr. Keller, is there anything else I can do for just a couple more points with an 89.49? Bryce, anything else that I can do? You've done everything you can. And if I gave him a couple extra points, is it fair to the rest of you? No, it's not. Okay, so I'm really trying to stick to these principles because I do feel it's the fairest for everyone. Okay? I'm not singling you out. It's not that I don't want to give you the grade. It's honestly that you just didn't earn enough points to get you to the point where it reflects as an A or as a B or fill in the blank, okay? I know that's a tough conversation, but do you remember the first day of class we kind of talked about that? Okay. So I've got this class. I will probably have grades wrapped up by the end of next week and formally post. I'll put an announcement saying, hey, grades are final. All right. I have office hours on Wednesday. I'll have office hours on Monday. If you have questions about your grade, you can email me. We can do our best to decipher it if you can't come in. But I really think you want to get a second set of eyes on your grades before you just decide not to show up on Monday. Okay? Eliana, review sessions for you this week.
Guys, so I'm going to be holding 2 exam review sessions this week. First one is going to be on Tuesday from 4 to 5 p.m. in Science Annex 117. And then the second one is going to be on Thursday from 5 to 6 p.m. in Science Annex room 122. And then Amara is also going to be holding 2 exam review sessions. Her first one is on Thursday from 7 to 8 p.m. in WETA room 242. And then her last one will be on Sunday from 3 to 4 p.m. in this classroom and also on Zoom. So as always, we'll give you out the participation code at the end. And more importantly, please come by and review for the final. And yeah, hope to see you all there.
Thank you. All right, so our average on exam 4 was 68 with our bonus. It was a little lower than some of the prior exams. Usually exam 3 and 4 are the more difficult exams. Typically the average on the last exam that's on Monday is our highest. It's just the way it works. I think it's because now we have a rhythm, you understand what to expect, the material is a little bit more straightforward, there's not as much of it. So if you're kind of planning and you're going to put in the energy, I think it's worth going for that grade that you're trying to get on Monday, okay? Why did the snake go to the baby toy store? To get a new rattle. Nicely done. When do astronauts eat lunch? Launch time, of course. Did you ever hear about that movie called Constipation? It never came out. It never came out. All right. Only 2 more classes where you have to deal with the dad jokes. Chemical senses. So we're going to be looking at, in this last unit, you've looked at, you watched the introduction online, and now we're diving straight into the material. So if you didn't see my announcement or you didn't watch that lecture, I think it would be highly advisable. It'll put it into context, what we're talking about this week. So it's just today and Wednesday. We're going to go through chemosensation today. We might get into hearing a little bit if we have some time. And then we'll finish out the rest of the special senses with balance and vision on Wednesday. So what's up here in the upper left? What is this an image of? This is a scanning electron micrograph of what? Any thoughts? Go ahead. It's part of the tongue. Yeah, that's well done. It's part of the tongue. It's called the valate papilla. Valate papilla that are on the tongue. Some students think it's a taste bud, but the taste buds are actually found in the crevices, and we're going to look at that. You probably remember this from lab. Did you guys already cover this in lab? Yeah. So, true to sensory input, the special senses are the way that we interact with our environment, right? But they're unique. They're unique. And in the introductory lecture, we talked about some of the differences between special senses and general senses. So somebody share with me what's unique about special senses compared to general senses. They're all located in the head. They're all here, right? There's no smell on your hands. Now, as an undergrad, I actually did research and looked at olfaction, or the sense of smell. And our model of interest was the tobacco hornworm, otherwise known as the Manduca sexta. And they have these really long antennae, and their antennae, which are still part of their head, their antennae are where their sense of smell is located. And that's true for a lot of insects. And that antennae go right to an olfactory bulb in their brain. And we're going to see that we have an olfactory bulb, and we bring in information on special senses, but it's all confined to the head. It's not like we're out here sniffing, around with our hands or seeing with our hands, right? Everything in the special senses is located in the head, but it has the same kind of purpose as general senses. That's to interact with our environment. So it's pretty critical for our survival. If we look at taste first, and then we'll look at olfaction. So taste and olfaction today. Both of these are chemoreceptors. Chemoreceptors, like we talked about in the online lecture. And the sense of taste, known as gustation, is a result of action from chemicals that bind to the taste buds. Now, these taste buds are located all around in the mouth, in the buccal cavity. That's what we refer to this as, the buccal cavity. So that could be the tongue, that could be the heart of the south palate, even on the cheeks. We see 4,000 taste buds mostly on the tongue and about 10,000 total that are located inside the cheeks, soft palate, the pharynx, that's like the back of the throat, and even the epiglottis. You can kind of see from this picture and appreciate that there are these bumps and these ridges, and you can feel that on your tongue, right? So these are the papilla. These are the papilla, and you've had like a dog or a cat lick your hand or your arm or your leg, and sometimes it feels really rough or like sandpaper. Well, they have specialized papilla that are responsible for that texture that you feel. And mostly because of the grooming activities that take place. But the different tongue papilla, lingual papilla, we've got filiform, folate, fungiform, and valate, or circumvallate as it's referred to. So let's look at these individually. The filiform papilla don't contain the taste buds at all. They're just for the texture of the food. You probably remember this when you were young, or if you have nieces and nephews. I remember when my kids were little. There was a rule in the house. They always had to try something. Okay, you had to try it. I got a funny story to digress on that. Well, one of our daughters really didn't like some of the foods that she ate. Like, she didn't like shrimp, she didn't like crab legs, she didn't like lobster. And my wife her mom would make special Valentine's dinners. And we would get all dressed up. Having 4 daughters, we'd get all dressed. They'd pick out my clothes. We'd all come to the dining room table, and my wife would make this really nice, like usually surf and turf of some sort, a really nice meal, but very cost effective, right? Because you buy it from the grocery, and every year we would force her to have a little bit of everything. She's tiny, like, 5 years old, 6 years old. One year, after having a little too much shrimp, she threw up. And she got super itchy. And her throat swelled and her eyes got red. Well, she has a shellfish allergy. And she would tell us, she didn't know that she had this IgE-mediated shellfish allergy. We got her tested the next week. But she would tell us that the texture is weird, Dad, really weird. But I tried it. I'm like, yeah, you tried it. And then she wouldn't finish anymore. But that was not a great dad moment, right? That was a pretty bad dad moment. So the filiform taste buds are there to try to help protect you. If something feels weird, especially with kids, they'll be like, I don't like this. Why? It tastes weird. What do you mean it tastes weird? It feels weird. The texture's off. And many of you have texture aversions, right? Certain foods are too slimy, right? You don't like your eggs a certain way. It's too slimy or whatever. So that's the filiform papilla that are giving you that information. The folate also has no taste buds, also giving you information about food texture. It's not as developed in humans as it is in other mammalian species, and it actually dissipates as you get older. The folate papilla is heavily present in young toddlers, and by adolescent years, they're pretty much gone, which also helps to explain certain like food aversions of kids saying, I don't like these certain foods, and then they're a teenager and they just eat anything, right? Maybe that happened to you. There were certain foods you just couldn't stand. And you're like, now they're really not so bad. There's one food that I've never liked, and I still don't like it, and that's broccoli. And the only reason I eat it is because my wife makes me eat it, but we kind of grow out of certain food aversions. I'm not talking about an allergy, like a true allergy. I'm not talking about that. But you'll see as time passes, there'll be certain foods that you used to not like that you try in a different social setting, and all of a sudden you're like, that's actually not so bad. Like, I don't know what the big deal was when I was a kid. Like, some of you are nodding, right? So some of this is because the anatomy and the physiology changes as we age. So folate. Folate is actually from folia, leaf, leaf-like. So they look more like a leaf. Fili is like a thread, so they're very thin, filiform. Folate are like broad, like a leaf. Now fungiform. Fungiform, what do you think they're named after shape-wise? Like a mushroom. Yeah, like a fungus. So they're at the tips and the sides of the tongues, the fungiform. They're widely distributed, but they're especially concentrated at the tips and the sides of the tongue. Fungiform. And then valate. Valate actually is a word that means wall. So these are at the back of the tongue, the rear of the tongue, and they contain about a half of all the taste buds on the tongue, towards the posterior aspect, like right before it leaves to go down the throat. So it's kind of interesting how this is distributed. It's interesting how these taste buds change over time, and the papilla adapt as well. So the taste buds really all look alike. It's the papilla that house them that have the shape differences. So now let's look at this figure. So up top is a valate, or a wall-like papilla, which is incidentally the picture that I showed on the first slide of the SEM picture. This is on the tongue. So the valate papilla, and you can see in these crevices, right here is where we're going to kind of blow up this magnification view down here. And you can kind of appreciate these little onion-looking structures, which is what this is. This is a large magnification view of a taste bud or a drawing. And these are the taste buds individually that are located down in this crevice. And we'll talk about the importance of that invagination or that crevice here in a second. This is actual histology of one of these invaginations or these crevices, and you can appreciate this taste bud sitting right here that it kind of has a look and feel like some of the receptors, the sensory receptors that you saw in skin. And that's what this is. It's a sensory receptor. But instead of responding to pressure or a stimulus, mechanical, it actually literally binds a chemical and gives you an action potential that tells you information about the chemical composition of that substance. And you interpret it in your brain as being sweet, and you're going to have another bite. Or bitter, like, I'm not sure I'm interested in that. It's a little too bitter for me. Okay, and that's really important. We're going to see here in a second of why that's important. So these are kind of, I think they look more like onions, but the textbooks call them lemon-shaped. So they're lemon-shaped groups of 40 to 60 taste cells with supporting cells. Now we saw this same motif like in the nervous system. Like the neuron is what gets all the attention, and the supporting cells were glial cells. Here, the supporting cells function like glial cells. They provide nutrition, but they don't actually send the signal. So it's the taste cells that actually do all the work. And so if you look at this bottom schematic or diagram, they're color coordinated. So the supporting cell is in blue, and then the sort of pink or purple looking cell is the taste cell. And there is a neuron with a synaptic vesicle. Isn't that interesting? Synaptic vesicle that will release a neurotransmitter onto the nerve, so the postsynaptic side, and send the nerve signal to the brain. But the supporting cells don't have any nerve innervation. They just support. They don't actually do anything else. So the taste cells have a tuft of apical microvilli. So these are like little hair-looking structures that serve as the receptor site to bind the taste molecule, or the chemical is what it is. So here's the taste pore. Here are the little taste hairs. And the taste pore is what houses the hairs, but this is literally where the chemical is going to bind. And then as the chemical binds here, that's going to trigger an intracellular signal, right? And you're going to release a neurotransmitter that crosses the cleft, then opens a sodium gate, and you get an action potential out the backside, just like we've learned about. So this is where we're kind of incorporating all of this stuff that we've already learned all semester. Okay, the basal cells. The basal cells, you can see, are located here at the back, the third cell type. We talked about supporting taste cells and now the basal cells. These are the stem layer that replaces either a supporting cell or a taste cell every approximate week to week and a half. Okay, so let's use a real world example. How many of you like pizza? Come on, raise your hands. I mean, pizza sounds good, right? After like Thanksgiving week, and you're kind of done with stuffing and turkey or ham, right? Pizza sounds good. How many of you get pizza and sometimes you move too quickly and put it in your mouth too fast? And you get a little pizza burn, right? As aggressive as it is, a few days later, it's pretty much gone, right? That numbness, because you replenish those cells that you just fried with that slice of pizza. Okay, thankfully, it's not like skin where it's like 6 to 8 weeks, right? We'd all be eating cold pizza if it was 6 to 8 weeks. But you can kind of get by, you got your nice slice of Fratelli's. You're going to hammer that sucker really fast, maybe too fast. You're going to be, dealing with it for a few days, but it might have been worth it. So the basal cells actually regenerate. These are the stem layer, like what we saw in skin. Very similar principle. And then these supporting cells, they resemble the taste cells without the taste hairs, synaptic vesicles, or sensory role. They literally just provide nutrition and support, like what we saw in the nervous system earlier this semester. So in order to taste, the molecules actually have to be dissolved, fully dissolved, and flood the taste pore. We're going to see the same motif happening in the nose. Because gustation and olfaction are both chemical senses, and gustation or taste and olfaction, smell, are closely related. In fact, you use them together. So in taste, we've got 5 primary sensations. You learned about this in lab: salty, sweet, sour, bitter, and umami. Salty, the interpretation of salty is when the chemicals or the food substance has chemicals that are high in sodium and potassium. These are produced by a lot of metal ions and gives that chemical signal of salty, which makes sense. Sodium, right? Sodium chloride is table salt. And so it tastes salty, sodium chloride, table salt, because it has a lot of sodium. Well, if you ate potassium, it would also have that same kind of flavor. Sweet, associated with carbohydrates in foods that have high caloric value. So you're interpreting the chemical sensation of high carbohydrates, high sugar content, as being sweet. Sour, acids in citrus fruits produce a sour type of flavor. Bitter is associated with spoiled foods and alkaloids like nicotine, caffeine, quinine, and morphine. So a lot of spoiled foods that are bitter won't taste nice, and you may not consume them. And that's a protective mechanism, right? And then umami is the meaty flavor of foods that are high in amino acid contents, like what you taste when you have chicken broth or a beef broth, even a vegetable broth. Okay, it's more of that flavor, that meaty flavor, or a meaty substitute type of flavor for those of you that are vegetarian or vegan. So this taste, though, is highly impacted by the texture. We talked about that. Highly impacted by the aroma or the smell. We'll talk about that next. Temperature as well as appearance. Right? I don't know how many times my kids would say, I don't want to eat that. Why? Because it looks gross. Like, what? What does that have to do with anything? Well, it has everything to do with it. Right? Your limbic system takes a look at that and says, I'm not sure. I'm not sure I'm putting that in my mouth. So the mouth feel or mouthful is detected by branches in the lingual nerve in the papilla themselves. And that's feedback that we get, mechanical feedback that we have on how much food you should actually put in your mouth in any one given time. I mean, you ever thought about that? I mean, there are social cues, right? You'll look, you're eating, you look over and you're like, oh, all right, and you got food coming out, and you're like, okay, maybe that was too much. So we get some social cues, but even still with infants, they're only going to put in so much food in their mouth because of this mechanical sensory feedback from branches of the lingual nerve that are found within the papilla themselves. Now, what's interesting, how many of you actually like spicy foods? Really like spicy foods? Okay. Now, spice is not a flavor. It's not on the list. So spicy is actually substance P, which is the neurotransmitter that gives you a pain signal. So if you like spicy foods, it's actually a pain signal that's coming to the brain that is providing you with some amount of pleasurable experience while you're eating your food. Isn't that kind of weird? But nociception is what's responsible for the detection of spice in a food. So when you go to your next Thai restaurant, you order spicy 5, right? You are a glutton for pain. You love it. That's what you're ordering. We have what we refer to as regional concentrations. And so a lot of times this is misunderstood, this map here. This is misunderstood. So the taste buds that are found in the papilla are located all throughout the tongue, all throughout the cheek, the soft and hard palate, the pharynx, even on the epiglottis. They're located throughout the entire buccal cavity. But there are areas of specialization that has higher concentrations of certain taste buds for certain types of flavors. What students inappropriately conclude from a drawing like this is you only can taste salty and sweet on the tip of your tongue. That's not actually the case. That's where the taste buds are the highest concentrated for those two sensations, but you can still detect sweet at the back of the tongue. It's just at the back of the tongue, it's mostly bitter taste buds that are found there. Okay? So sour, you can kind of tell, like you get this sour feeling, and it's like the side of your tongue kind of lights up when you have some sort of like sour, gobstopper or something, a sour piece of candy. So these are regional concentrations, not to be confused with this is the only place that they're found. Okay, so let's talk physiologically or mechanistically about taste, and then we're going to quickly move into olfaction, or the sense of smell, because they're intimately tied. And I'll give a couple examples of that. So we've got sort of two mechanistic processes of action that can take place here. So either we can use, you've seen this motif before, like when we looked at neurotransmitters, right? We had metabotropic or we had ionotropic. You guys remember that? It's the same theme here. So if it activates a second messenger system, it behaves a lot like a metabotropic neurotransmitter. And the second messenger system sends a signal inside the cell, some metabolic process within the cell, to then release the neurotransmitter on the basal side to stimulate the nerve. And the flavors, if you will, that operate mechanistically through a second messenger system are sugars, alkaloids, and glutamate bind to receptors which activate the G proteins. So sugars are sweet, alkaloids are bitter, and glutamate are the umami flavor. So sweet, bitter, and umami all are through a second messenger system. That leaves sodium and acids. Sodium, salty. Acids are what? Sour. Acids, sour. Sodium, salty. Depolarize the membrane directly. This should be easy to remember, in my opinion, especially for salty, because it has sodium in it, and that's going to depolarize the membrane instantaneously. So either mechanism releases these neurotransmitters on the basal side. They bind on the postsynaptic side of the cleft, and they trigger an action potential to be propagated to the brain. Okay, so lastly, and I promised you this, I know in lab you memorized these. There's really only three that I want you to focus on here: the facial, the glossopharyngeal, and the vagus. So the facial nerve, which is also cranial nerve what? 7. The facial nerve collects sensory information from taste buds over the front 2/3 of the tongue, sending it back to the brain. The glossopharyngeal nerve covers the posterior or the back 1/3 of the tongue. That's cranial nerve 9. And then the vagus, cranial nerve 10, taste buds from basically everywhere else: palate, pharynx, and epiglottis. And this is how you're getting your information about taste, through these cranial nerves that go straight to the brain. So from an anatomical location, think about this for a moment. How important is taste to us as a species? If it's located in our head, it has 3 direct cranial nerves that take the information straight to the brain, right from here. Right? I mean, it's critical for our survival. Like, you have to consume calories. You have to find calories. You have to find foods. Like in early human development, you had to find foods that had a high caloric value, like a lot of carbohydrates, if you're foraging. Right? And then you need more protein as humanity evolved. And so we would look for more umami flavored, and that gives you sort of this satisfied feeling of being full. is that umami flavor, right? Like a candy bar only does so much. But like a ham sandwich, you're like, whoo, that tasted good. Okay. Questions over taste before we kind of just swing into olfaction or the sense of smell. Now, these are intimately connected. How many of you were sick this semester? Okay. How many of you had a head cold? Okay. How many of you had a head cold in the past and know about what I'm going to say? What does food taste like when you have a head cold and you're all plugged up? Blah. That's exactly right. You can't really taste anything. Right? Well, how many of you had COVID? Did you lose your sense of smell? Yeah. We'll talk about that here a little bit later, but kind of interesting, right? So when you just have the regular old cold, skip COVID for now, just think about the regular old head cold. I'm not talking about a respiratory chest thingy, or a stomach thingy. I'm talking about you're congested, you've got a lot of phlegm, your nose is plugged up, your head feels like it's gonna, explode. You're having a difficult time bringing chemicals to the olfactory epithelium for them to bind. Your tongue's working just fine. So arguably, olfaction gives us more cues about taste than even gustation. Okay, so let's play this out. What was the biggest number that we had of taste cells in the last part of the lecture? 6,000, 10,000, right? So we're in the thousands. The olfactory mucosa contain 10 to 20 million olfactory cells. We are multiple orders of magnitude greater than what happens in the mouth, right? If I gave you $5,000, you'd be like, sweet, thank you, Keller. Best professor ever. But if I gave you $20 million, you'd be like, this guy rocks, right? Rate my professor off the hook. I mean, orders of magnitude difference. So no wonder when we have a head cold, you can't taste anything, and your tongue is working just fine. Maybe the back of your throat's a little phlegmy, but your tongue is fine. It's your nasal epithelium that are all plugged up. That's why you can't taste anything. So similar organization here. We've got supporting cells, we've got olfactory cells, we have basal cells. But the mucosa of the superior concha, nasal septum, and the roof cover about 5 square centimeters. So 5 square centimeters, that's about 5 centimeters there. So 5 square centimeters is like this, from here to here, from here to here. So it's about like that. Right? And everybody do this. Put your thumb and your pointer finger together. That's about 5 square centimeters. Now put that up to your face. That is not the size of your nose. Okay? I don't care how self-conscious you are about your nose. Your nose is not this big. Okay? That much epithelium full of olfactory cells you've wrapped and stuffed up in here. Folded, right? Huge amount of chemical sensation is right here with our sniffers. And honestly, the size of your nose has nothing to do with it, right? It's all really internal. Oops, I got excited or something. Okay, so on average, well, not on average, but how many different tastes can we detect with the tongue or the buccal cavity? 5. You can distinguish 2,000 to 4,000 different odors. So much more colorful, if you will. Right? It's literally almost like black and white versus color TV when you're talking about smell versus taste. So where are we? Well, look at this. This is the olfactory mucosa that's sort of like peeled away. These little green spheres are odorant molecules that are binding. And this is the olfactory bulb right here. we're gonna take a look at this here in a little bit more detail. But what's interesting is this is one of the few places, the only place in the body where the neurons are directly exposed to the outside environment. Now, it's kind of protected because, you've got this structure that covers it. But essentially that airflow and those molecules are interacting directly with the nerve. Directly with the nerve. How many of you have ever been pepper sprayed? Yeah. Any Michigan fans here? No, you doesn't know what I'm talking about? Okay. You could Google pepper spray and Michigan fans and you'll see something come up. It's kind of fun. Yeah, they like slapped him on the hand with like $100,000 fine. That's like nothing, right? I think the water boy probably makes that. University of Michigan and Ohio State University, those programs are very well funded. I don't think $100,000 is going to do anything. If you guys don't know what we're talking about, they played each other over the holidays, and Michigan upset them, and it was at OSU in Columbus. And then one of the Michigan players brought the flag to midfield and tried to like plant it. And they got in this big brawl, and the police had to pepper spray people. But anyways, that's why it burns so much, because you have direct impact to the nerve in the nose when pepper spray is being released. Okay, so these olfactory cells, they're neurons. They're actual neurons. They're shaped like little bowling pins. The head themselves has 10 to 20 different cilia or olfactory hairs, very similar to the taste hairs that we just got done looking at. And they have binding sites for an odorant molecule, a chemical. That's what that means. An odorant molecule is just a fancy way of saying a chemical. But these hair cells are not modal, right? They literally serve the purpose of binding a chemical. And they lie in a layer of mucus. Why? Why do they lie in a layer of mucus? Give me some reasons as to why. What's important about detecting a chemical from taste? Has to be dissolved in saliva. You don't have saliva in the nose, but you have mucus. Same principle. Okay? So one of the purposes of mucus in the nose is to dissolve the chemical. Give me some other purposes for having mucus in the nose. Trap the molecules as they go by, right? You're like, right? You're trying to smell. What else? To stay moist, absolutely. Anything else? These are bare neurons. Protection. Absolutely. So when you have a cold, you have a nasal irritation, mucus production goes up, right? It's a compensatory mechanism to protect your nasal epithelium. Now there's a fine balance between having too much mucus and not enough mucus, right? Then you lose the sense of smell. Protection. We're gonna kind of introduce it, but you'll cover it next semester in respiratory. But one of the purposes of the nasal concha is to actually warm the air, moisturize the air, and to trap contaminants or particles so they don't go into the lungs, right? And that's why we have mucus. That's why you have boogers, right? That's just debris that's in the air that you've trapped before it made it into your lungs. So if you think boogers are gross, just be grateful that that's not getting into your lung. Like your body is trapping it before it makes it into your respiratory epithelium. So on this picture, just to lay this out, this is airflow. This right here is the cribriform plate of the ethmoid bone. You studied that in lab. There are holes that are naturally occurring because this is the pathway by which the neurons actually extend to the brain. This is the brain. This is the olfactory bulb of the brain. And an extension of that through the ethmoid bone gives rise to the olfactory cell. Here's the olfactory cell. Right next to it is a supporting cell. And then the basal cell is towards the basal epithelium. That's the regenerative layer, just like what we saw with the tongue. This odorant molecule is going to bind to these olfactory hairs, synonymous with taste hairs, and is going to trigger an action potential to come send a signal onto this second order neuron that ultimately sends the signal back to the brain. And that's how you smell. So a couple of things that are really important with smell, right? How many have ever done wine tasting or seen this happen, right? Well, very well-trained individuals can actually detect the taste of the wine without even putting it in their mouth. And you're like, what's the point? I kind of agree with you, but right? So they swirl it around. They sniff it first. They determine certain attributes about the wine based upon how it smells, even how it looks in the glass. And then ultimately they might palatize it, put a little bit in their mouth, move it around so it lights up all these different gustatory cells. And then what do? They literally discard it. They spit it out. I think they missed the best part. But, I mean, you can see why this process has been so refined, right? Same thing kind of happens with different types of food tasting, except for the spitting out part. But there's a lot of smelling. There's a lot of texture. If you watch any cooking shows, they talk about the texture, right? So there's so much of this anatomy and physiology in this world that we live in of taste and smell. Okay. So to continue, the basal end of each cell becomes the axon. The axons collect into small fascicles and they leave the cranial cavity through this cribriform foramina, the holes. And the fascicles are collectively referred to as cranial nerve 1. Cranial nerve 1. Cranial nerve 1 is referred to as what? The olfactory nerve. So this is the one place in the body where we've got neurons directly exposed to the outside environment. They only have a lifespan of about 60 days, 2 months. And the basal cells, the regenerative layer, divides and differentiates into new olfactory cells. And we kind of talked about the role of the supporting cells and the basal cells. Okay, so a couple of sort of pieces of fact. So we actually have one of the worst sense of smell of any mammal. And I actually think it's getting worse as time goes on. Because I think we're lazy, right? We've been able to become lazy. I mean, what do you do when you buy something or soda, or you're like, what flavor is it? You go, oh, it's cherry. Look, it says it right here. Like, we don't even bother, like, what flavor is it? It's grape. I mean, you just read it. So women do have more sensitivity to certain odors than men. Smell is very highly important in a lot of social interactions. We talked about different types of sweat glands. Remember this? This is our dog. This is Nori. She's a Portuguese water dog. But like all dogs, what's one of the first things when somebody new walks into your house, your dog does? Smells where? their crotch, right? Their butt, their crotch, right? Like, some of you are like, yeah, it's just because that's the closest thing to them. No, that's where so many of those sweat glands that give off an odorant that is distinct to you. The bacterial flora that live on your skin are unique to you. You have a unique smell to your dog versus anybody else. Your gym shorts, your stinky gym socks smell wonderful to your family dog. Okay, and they know that it's you. And if you've been away at school and you went home for Thanksgiving and you have a family pet, they probably knew you were home. And it was mostly because they smelled you. Right? Dogs on average have 100 million more receptors in their nose than we do. We talked about 4 to 10,000 in the buccal cavity of chemosensation. Then we talked 10 to 20 million in our nose. Now we're hundreds of million above that for dogs. Bloodhounds have 300 million more. Right? Every different strain or species of canines has different capabilities. So the odorant molecules are going to bind to membrane receptors on the olfactory hair. If they're hydrophilic, they'll diffuse straight through the mucus. Hydrophilic means water-liking. If they're hydrophobic, they have to be transported by an odorant binding molecule. Then they're going to activate a G protein in a cyclic A&P system. That's a second messenger system inside of the cell. And that opens the ion channels using sodium or calcium and depolarizes the membrane. So there is no direct depolarization like what you saw in taste. That's one of the big differences. It's using a second messenger system with olfaction. So the action potential goes to the brain. The olfactory receptors, they adapt actually quite quickly due to synaptic inhibition. So this adaptation that takes place, we have in our first lecture we talked about this, like phasic was adapting quickly and tonic is adapting slowly. So this adaptation that takes place, like the analogy I think we talked about in that lecture was, you ever walked into a room and maybe you came home for Thanksgiving and mom or dad or grandma and grandpa were already cooking. You walked in, you're like, wow, it smells good. Well, 10 minutes later, if you took a check, you wouldn't really notice because you've adapted to the sense of smell. But if you went out in the backyard and played with your dog and then you came back in, you just reset it. So this adaptation that takes place happens very, very quickly. Some odorants act on nociceptors of the trigeminal nerves, like what you will find in pepper spray, right? Or mace, like the analogy, the recent University of Michigan OSU game. So things like ammonia, menthol, chlorine, capsaicin of hot peppers, these actually bind on nociceptors. Those are pain receptors of the trigeminal nerve. Now, true to form, I promised you would kind of come back to just a little bit on COVID, but COVID's not alone. I mean, there's a whole class of viruses that can cause these nasal and sinus congestion, and they can interfere with the molecules reaching the receptors, like what we talked about. But in some cases, the virus itself, like in COVID-19, actually damages that olfactory sensory neuron, and it can cause trauma at such levels through inflammatory processes where it's unreversible. And that can be lost either forever or for a long period of time. Because you're not talking about the olfactory cell, you're talking about the nerve itself. Okay, questions? What I'm going to do, because we have a little bit of time, so we have a little bit shorter lecture on Wednesday, I'm going to go ahead and start hearing anatomy. We won't get into the physiology. We're just going to start the hearing anatomy. Stay with me, because I'm going to give you a code at the end of today for one of our participations. Okay, so hearing and equilibrium. We won't get to equilibrium. We're just going to briefly introduce hearing, and I'm going to try to find a nice spot here that I kind of pre-selected for us to hit the pause button. So we come back on Wednesday for our last lecture. We don't have as much to do. Fair? So hopefully you're in agreement. Hopefully I've convinced you that the sense of smell and the sense of taste are critically important for our survival. We use them far more than you realize. And when they're gone, like in the case of like a COVID-19 infection or just a head cold that got really bad, when they're gone, you really miss them, right? There's nothing worse being sick over the holidays, right? And you've got, maybe some home cooking that you're looking forward to or a grandma recipe, and you're like, it just doesn't taste the same. Right? Now, true to that form, hearing equilibrium, hearing and balance, as well as vision, are just as important to our daily life. Right? Hearing, this is a response to vibrating air molecules. So these are mechanoreceptors. We just left chemoreceptors, and now we're moving into mechanoreceptors. There's a mechanical response. Equilibrium or balance is the sense of motion or body orientation. And you're going to see there's also that's also mechanosensation here as well. Both of these reside in the inner ear. And this is a maze of fluid-filled passages and sensory receptors. The fluid moves. The sensory receptors convert this motion into a pattern of electrical signals, action potentials, like what we've been learning about. So this is a very simple diagram to describe kind of the three areas of the ear. And then we'll show a more anatomically relevant one as we kind of wrap up today. So on your left, you can see the outer ear. The outer ear is just really to capture sound waves. And sound waves are just that. They are waves, mechanical waves, sound as it pushes through a medium. The medium that it's pushing through right here is air. Right? And you all know, like if this was a vacuum, well, we'd all be dead. But if we were living in a vacuum and we could survive, sound has nothing to push in a vacuum. So in space, there's no sound. There's no perception of sound. And I remember as a kid when the neighbor down the street explained that to me, the 2-year-old neighbor, and told me that Star Wars was fake because there were really no sounds in space. And I was like so crushed. But it's true. Now, the other thing is you can experiment with this. How many of you have been swimming in a swimming pool? And you ever play this game where you're shouting at people in the water, underwater? You can still hear, but it's like, right? Because the medium is different, and the sound waves that are moving through a thicker, more viscous medium. Okay? Because we have an atmosphere, we can actually hear. So that is the purpose of the outer ear. Now, the outer ear has this border to the middle ear by this eardrum known as the tympanic membrane. And as the sound waves come through, the eardrum or the tympanic membrane actually vibrates. It vibrates and it moves 3 bones in the middle ear. 3 bones that make up the middle ear. Those 3 bones move. They push on another membrane at the oval window. And on the other side of that inner ear boundary is another fluid. And this is a liquid versus air. Now you see this auditory tube that connects from the middle ear basically to the back of the throat or essentially the nasal pharynx. What's the purpose of this opening right here? Auditory tube, otherwise known as the eustachian tube. What is it? Pressure regulation is the main thing. Drainage when there's an infection. That's right. How many of you had lots of chronic ear infections when you were kids? Okay, how many had tubes in your ears? This is where the tube went to make sure this stays open. That's where the tubes were implanted. A very, very common procedure. Okay, so if we look at in all of the physiology happens in the fluid, and we're going to talk about the physiology Wednesday. Okay, we're just setting up the anatomy. So, what is sound? Well, I kind of talked to you that sound is a mechanical vibration, right? Vibration of air molecules. It could be water molecules. In the inner ear, it's going to be a perilymph or an endolymph. That's the fluid that's there. And there's a couple of components of sound, like pitch, whether it's treble, which is high, or bass, which is low. This is measured in frequency. So high frequencies are treble, like 7,000 to 20,000 hertz. And hertz is like a cycle per second. Cycle per second is what a hertz is. And then low frequency or bass sounds, low frequency, is low numbers like 20 to 800 hertz. And these numbers have become important because you're going to see when we get to the physiology that the bass and the treble are detected in different spots. And as we get older and we lose our hearing in specific areas. Or you could have the loudness aspect of a sound. This is the intensity, the amplification or the vibration. This is measured in decibels. Okay, so here's a picture that's more anatomically relevant, but it has a lot of the same parts. Here's the outer ear, and its border is the tympanic membrane or the eardrum. The middle ear houses the three bones, the malleus, the incus, and the stapes. Some other terminology is hammer, anvil, and stirrup. These happen to be the three smallest bones in the body. And they're literally connected. And when this vibrates, these three move in unison and pushing the stapes on that oval window. Now, this snail-looking structure, right? But before we get there to the snail-looking structure, here is this auditory tube right here. But once we pass the stapes and the oval window, we're into this snail-looking structure, which is all considered the inner ear. And this is housed within the skull. This is all bony architecture in the skull. This can be torn or ruptured, and it can heal to some extent. Not always. How many of you have had kind of a torn eardrum or known somebody? Yeah, so it can be pretty painful, but it will heal over time. If it's aggressive, it might have to have surgery. These bones are known to undergo arthritic conditions or osteoarthritis. So this is one of the potential culprits of losing hearing as we age, is kind of the mobility of those three bones. But then most of the physiology is found in this snail-looking structure that we're going to look at. Now, the back of the snail, this is called the cochlea. This is actually where you hear. And then the top part, which kind of looks like, above the head, if you will, of the snail, this is the semicircular ducts. So these are the vestibular apparatus. This gives you information about balance. This is the cochlear apparatus. This gives you information about hearing. Out of the cochlear apparatus, it's innervated by the cochlear nerve. The vestibular apparatus is innervated out of the vestibular nerve. They come and they fuse and they meet, making up the vestibular cochlear nerve. Make sense? Inside of the cochlea, we're going to take a look at that on Wednesday. We'll open up some of this architecture of the semicircular ducts here momentarily, but we're going to look at that physiology in greater detail when we look at the lecture on Wednesday. So the cochlea. If you take this structure and you unwind it, you can see that there is a top portion, there's a bottom portion, and then there's a middle portion. And each one of these is made up of a fluid. So it's scale of vestibuli on top that has perilymph that's colored in green, scala tympani on the bottom with perilymph also colored in green, and then the cochlear duct in the middle, which is actually made up of endolymph and is colored in blue. Within the cochlear duct is this organ, this structure, this diagram in rectangle is blown up over here. This is our hearing apparatus. This is the tectoral membrane sitting on top. This should look somewhat familiar. We've got nerves coming to cells that have hairs, just where we were. But these hairs don't bind an odorant molecule. These hairs are literally inserted into the tectoral membrane. So, how this works. Air molecules vibrate, the eardrum moves, the three bones move, the oval window moves. On the other side of the oval window is this fluid-filled chamber, and it propagates the wave of sound through the fluid. Like when you take a pebble and you drop it onto a pond that's nice and calm, you can literally see the ripples. Those ripples flow through these different chambers, moving the tectoral membrane, right? It's like that. That's like the raft that's sitting on the top of the pool, is the tectoral membrane. And when it moves, the hair cells detect it, and they transmit the signal of mechanosensation. So you're literally mechanically detecting the sound. All sorts of things that can go wrong here, right? We talked about the eardrum. Now we're inside here. Well, what about these hair cells? Can they break? Can they dissociate with the tectoral membrane? Can you actually have the nerve detached from the back of the hair cell? You see there's supporting cells here. There's a basal membrane. Where are the basal cells, the regenerative cells? Where are they? They're missing. Why? I don't know why they're missing, but what does it mean? It means that it's non-regenerative. So if you damage a hair cell, there is not a basal cell like what we saw in olfaction or gustation to recover from the pizza burn, for example. Okay? So protecting our hearing, that's why we take great lengths to protect our hearing. Right? And a lot of jobs that have high sounds and they measure it in decibels and they'll drop it below a certain limit. This is why. Because there are no regenerative cells within the inner ear. Okay, so this is just kind of filling out the rest of the information that we kind of talked about. Hair cells and supporting cells. This organ that I refer to is referred to as the organ of Corti. That's the hearing organ. Now, what's interesting about perilymph is the green-colored one. It's in the scala vestibuli and the scala tympani. In the perilymph, the main ion that's present there is sodium. But in the cochlear duct, where you have endolymph in blue, potassium is the main ion that's present here, potassium ion. So when we look at the physiology on Wednesday, I'll remind you of that, but you're going to see some unique differences from what we've seen with an action potential, because it's actually potassium's turn versus sodium's turn. Okay? Questions? We good? Finish a few minutes early. We'll have a few minutes early finish on Wednesday as well. Stay put for 1 second.